Hello. I have created a presentation examining the hormonal control of implantation and early embryonic development in pregnancy with a focus on the importance of communication and coordination. Pre-implantation. First off, it is essential to know that implantation refers to the process by which an embryo attaches to the endometrium of the uterus, eventually invading it to meet with the mother's circulation and form the placenta. However, before this key event occurs, conditions must be perfect in the uterus, which is where the extremely important factor of hormonal control comes into play. As you can see in the diagram to the right, the corpus luteum is a yellow fatty structure that forms temporarily in female ovaries from the rupture of the ovarian follicle that released a mature egg during ovulation. The corpus luteum functions as an endocrine structure before the formation of the placenta in the case of fertilization. It is the corpus luteum that ensures that pregnancy begins and that conditions in the uterus are properly maintained to support a growing human. In terms of hormonal control, upon implantation, which we will discuss further in the next slide, specialized cells of the embryo secrete human chorionic gonadotropin, also known more simply as HCG, which is a peptide hormone that essentially rescues the corpus luteum and signals for the maintenance of progesterone secretion. Progesterone secretion in these very early stages of pregnancy allows for the endometrium to be maintained to support growth and development of the embryo, as well as to suppress uterine contractions. Additionally, the corpus luteum secretes the hormones called estradiol and inhibin A, hormones that contribute to development of milk-secreting ducts and provide negative feedback control of follicle-stimulating hormone, or FSH, secretion from the anterior pituitary gland, respectively. Okay, stages of implantation. Now that we have discussed the essential hormones of pre-implantation, we can begin the discussion of the 4 stages of implantation. The first step of implantation is called hatching. Hatching is the name of the process of the loss of the zona pellucida, which is a layer of glycoproteins that surrounds the plasma membrane of human oocytes and serves to protect premature implantation in the fallopian tubes. The next stage is called apposition, during which the blastocyst loosely contacts the implantation site in the endometrium of the uterus. This site is typically located in the upper and posterior wall in the anatomical mid-sagittal plane. This step is mediated by an increase in a specific type of prostaglandin that functions to regulate trophoblast invasion, activating other signaling proteins. Finally, during this process, the blastocyst differentiates into an inner mass of cells that will eventually become the embryo, and an outer layer that will become the placenta called the trophectoderm. In fact, in the third stage of implantation, termed adhesion, the blastocyst trophectoderm adheres to the lumen of endometrial epithelial uterine cells through mediation by cell adhesion molecules, including uterine pinnipod protrusions, as you can see in the diagram. The hormone progesterone enhances their development, while estrogens inhibit their development. In the final stage of implantation, called invasion, fetal trophoblast cells are allowed to invade the endometrial epithelium and enter into the endometrial stroma, which has a rich blood supply from the spiral arteries, which are reconstructed into chorionic villi that contain embryonic blood vessels and will eventually become the placenta. Okay, post-implantation. Now that we are familiar with the stages of implantation, we can take a look at the key hormones of post-implantation events. Implantation occurs on average 8 to 9 days after fertilization. As we briefly talked about earlier, human chorionic gonadotropin, HCG, is instrumental in promoting the release of progesterone by the corpus luteum. However, it is important to point out that progesterone, and estrogen for that matter, is produced continually throughout pregnancy. Secretion of HCG, often called the pregnancy hormone because it is detected by blood and urine tests to initially detect pregnancy, typically peaks at 3 months of pregnancy and then diminishes because the placenta secretes progesterone as an endocrine structure once it is fully formed. It is after week 8 of conception that the embryo begins to be referred to as a fetus. The placenta develops soon after the fetal period begins, around week 10 after conception on average. The development of the placenta is a major turning point for hormonal control of pregnancy, as the placenta functions as an endocrine fetal-maternal organ. In addition to the secretion of progesterone and estrogen, the placenta secretes numerous other important hormones that protect the growing fetus. The placenta secretes a peptide hormone called human chorionic somatomammatrophin, or HCS, which namely increases breast development during pregnancy and alters the mother's glucose and fatty acid metabolism to support fetal growth. The placenta also secretes a number of hormones to promote the tremendous amount of growth that occurs during fetal development. The placenta secretes a peptide hormone called insulin-like growth factor, which has been found to have a positive correlation with fetal birth weight. It promotes fetal growth by stimulating placental growth, increasing both nutrient transport and delivery to the fetus. Next, placental growth hormone replaces maternal pituitary growth hormone and is secreted continuously into only the maternal circulation and serves to stimulate gluconeogenesis, lipolysis, and anabolism in the maternal liver as well as in her other organs. Moreover, in an indirect manner, placental growth hormone increases nutrient availability for the fetus. This hormone is important during the end of pregnancy, where growth of the fetus is especially important. Vascular endothelial growth factor acts as a paracrine hormone on fetal placental circulation and increases vasodilation, which effectively increases fetal nutrient supply, making it essential for placental and fetal growth and development. The final placental hormone that I think is important to mention is called relaxin. Relaxin is a peptide hormone similar to insulin. This hormone softens the cervix and the birth canal tissues in preparation for parturition, and estrogen further enhances these effects. Additionally, it has been found to promote the growth and differentiation of glandular, fibrous, and supportive mammary tissues to prepare for lactation. Pathologies. Unfortunately, there are various possible pathologies that can arise with implantation and early embryonic development, including ectopic pregnancy. This arises most commonly when implantation occurs in the fallopian tube. 94% of ectopic pregnancies are tubal, but they can also occur in other inappropriate places, such as the ovary and external surface of the uterus. Many times, this happens if the zona pellucida is lost too early or if uterine epithelium is damaged. Early abnormal levels of HCG can be used to diagnose this condition that cannot produce a viable fetus. Another pathology of this type that can occur is called molar or hydatidiform pregnancy. This occurs when only the trophoblast layer grows while the embryoblast does not. Therefore, no embryo is able to develop. This can cause a tumor to form within the uterus, but as the trophoblast cells will still secrete HCG, many times this condition is not diagnosed until an ultrasound is performed.